Okay, I'm going to go ahead and start then. Good morning, everybody. I'm Marco Ferrara. I'm one of the colon and rectal surgeons down in Orlando. I am affiliated with our colorectal fellowship program here in Orlando. I may have met some of you during the interview process. So I'm going to be giving 3 lectures this morning. The first one we're going to do is pretty wide-ranging, but it's colorectal cancer, the genetic background of that, and also a little bit about colorectal cancer staging. This is a very highly tested topic on the written and oral boards, very important. And so we'll just kind of go over some of the basic science behind it, and then, the kind of modern staging of this. We won't get too much into the treatment itself. There'll be kind of separate lectures for that. So these are our objectives. So we're going to talk about the molecular basis of colorectal cancer, talk about some of the inherited syndromes. We're going to talk about colon and rectal cancer staging. So this is from our ASCRS textbook. So some key concepts: colorectal cancer is caused by the accumulation of a variety of genetic alterations in colonic mucosa. It can be hereditary or sporadic, not inherited, and both forms may share many of the same genetic alterations. There's multiple hereditary forms that have an expected phenotype due to the genetic alteration that increases the likelihood of the cancer. And there are many screening algorithms for colorectal cancer, and these differ between hereditary and sporadic cancer based on the time expected for an adenoma to become a carcinoma in that patient based on their genetic makeup. And hereditary forms of colorectal cancer are more commonly seen in patients with young onset colorectal cancer. And there are treatment algorithms for hereditary colorectal cancer, and these are directed towards removal of the cancer, as well as decreasing future risk of additional cancers. So let's talk about the epidemiology of sporadic cancer. So colorectal cancer is the 3rd leading cause of cancer-related death globally. 80% of colorectal cancer is considered sporadic, meaning that the genetic alterations in the tissue versus germline when inherited. There's a pretty much even distribution of male to female. Most patients are over 65 years old, although we are seeing increasing incidences of colon and rectal cancer in the younger population. This is part of why recently multiple associations have recommended to move up the colon screening guidelines to a colonoscopy at 45 years old. It's most common in the right colon, 41%, or in the rectum, 28%. Some of the risk factors: genetics, lifestyle, dietary, environment, these all play some part in increasing the risk. There's about a 12% increase for every 100 gram per day increase of red or processed meat. Alcohol has also been shown to increase your risk of colorectal cancer. Whole grains and dairy are found to decrease. The mechanism of action is unknown. It may act via microbiome changes. There's some idea of maybe some microinflammatory processes that over long periods of time can lead to increased cell turnover and possibly increased risk of cancer. This is an area of a lot of study currently, trying to find other ways to mitigate the risk of colon cancer. Also sedentarism and obesity. So let's talk a little bit about the molecular basis. So sporadic colorectal cancer molecular genetics. So there's a lot of different pathways through which colon cancer can arise, that is, in a sporadic way. So there's genetic alterations. You can have accumulation of copy number variation. There's CIMP, which is the CpG island methylator phenotype. There's also kind of the more traditional pathways we think about via APC, P53. You can have changes in your, so a lot of these different kind of ways that we kind of get. This is a little bit more than what we would probably be tested on, but just kind of goes to show there's a lot of different genetic pathways, and our understanding of this has really grown recently to where we're able to maybe target and identify high-risk candidates a little bit better for maybe even targeted treatment later on down the road. So in general, you can, from a molecular biology standpoint, divide this into hypermutated versus non-hypermutated tumors. So hypermutated has over 1,000 different mutations in a cancer. And we kind of go, this is kind of the general idea here of how a cell will become cancerous. So you have evasion of apoptosis, you have self-sufficiency in growth signals, insensitivity to anti-growth signals, tissue invasion and metastasis, limitless replicative potential, and sustained angiogenesis. These are kind of the cycle here that allows a normal tissue to become cancerous and invasive. So this is from a study that has been referenced to quite a bit, but so they kind of define 6 alterations in regulatory mechanisms. So kind of like what you just showed in that image. So self-sufficiency and growth signals. So cancer cells must lose this need of exogenous growth signals to proliferate. There's insensitivity to anti-growth signals, so growth inhibitory signals to maintain cell in quiescent states, invading apoptosis, limitless replicative potential, and sustained angiogenesis. So they must develop the ability to induce and sustain angiogenesis. Otherwise, it'll outgrow its blood supply and become necrotic, and then also develop the ability to invade. And the sequence of these events and mechanisms, it varies. So you can have, again, there's oncogenes. These mutations result in abnormal gain or excess of a particular protein function. This is like the RAS, the c-MYC, the CERB2. And then there's also tumor suppressors. So you can have inactivation of tumor suppressors, which normally inhibit cellular proliferation or promote apoptosis. That's APC, DCC, P53, MCC. This is kind of a very traditional pathway that we see. Normal epithelium, usually APC mutation, will then lead to adenomatous changes. Many tumors will then have a KRAS mutation, and then eventually P53 mutation. These are found in a large majority of colon cancers. So the RAS oncogene, so this is the most frequently mutated in CRC. The KRAS proto-oncogene is on the short arm of chromosome 12. It's mutated in 50% of colorectal cancers. The KRAS gene product involved in transduction of exogenous growth signals. Chemotherapy is, this is important in chemotherapy. You check for mutation. If it's KRAS positive, you can't give certain medications like cetuximab. It has a poor overall response. And again, c-MYC and CERB2 are other oncogenes that have been identified. Then there's tumor suppressors. So again, they normally inhibit cellular proliferation or they promote apoptosis. And when they're lost, you have loss of the normal inhibitory control of cell cycle. And it's the Knudson's 2-hit theory. So it's inherited, somatic, or both. And this is kind of showed here. You have one copy that's mutated, and the second hit is what actually leads to the loss of function. And again, this is just showing that same pathway here. So the APC gene, this is located on the long arm of chromosome 5. It's considered the gatekeeper of carcinogenesis. There's a mutation in APC, appears to be the initiator of disease, and it's found in 50% of sporadic adenomas and 75% of sporadic colorectal cancer. The APC protein it normally regulates the WNT, the wingless signaling pathway. There's a tumor suppressor gene, and when it's inherited, its cause is FAP. And it also causes increase in beta-catenin, cell signaling proliferation, and cell-to-cell adhesions. The P53 gene, this is located on the short arm of chromosome 17. It's most commonly mutated gene in human cancer overall. It slows the cell cycle. It facilitates DNA repair during replication. And when not feasible, it is what helps to induce apoptosis. An inactivation of P53 is found in 75% of sporadic colorectal cancer, just like AFP or APC. They are not the initiators of colorectal cancer, usually. There are some diseases like Li-Fraumeni, which is an inherited defect in P53. Those patients do not have an increased colorectal cancer risk, although they have many other increased risk from a cancer standpoint. And studies show lower survival rate when P53 is negative or mutated compared to tumors that express P53 product. So another is deleted in colorectal cancer, DCC. This is a gene on the long arm of 18q. It's mutations found in majority of colorectal cancer. A product of DCC is a transmembrane protein important in cell-to-cell adhesions. There's inactivation of DCC enhances metastatic potential. And similar to P53, its positive tumors have better prognosis than DCC negative or mutated tumors. Then mismatch repair system. So MMR genes, they act as basically spell checkers. They'll detect errors in DNA coding and will correct those errors during replication. If you have a base pair mismatch, they'll find them, they'll excise them, they'll correct them. And lack of this can lead to an accumulation of errors, which then can lead to defects of DNA replication, defective proteins. And these things are identified by microsatellite instability. So these are small regions of DNA in the genome that do not code for individual genes as far as we know. We don't really know exactly what the function of these islands of kind of non-coding DNA are. And their small base sequences are different in everyone. They're susceptible to MMR gene defects, and this is termed microsatellite instability. The NCI recommends tests for 5 microsatellite sequences to test for MSI, and 15% of colorectal cancers demonstrate MSI. I think it's pretty standard now in most hospitals for this to be checked on pretty much any biopsy or any tumor that's removed. They'll check for MSI status. So MSI high means that 2 or more of the 5 demonstrate microsatellite instability. These tumors are more likely to be high grade, more likely to be right-sided. If it's MSI low, then you have 1 of 5 versus MSS, which is microsatellite stable, which means that none of those have been knocked out. So MSI tumors have better prognosis than microsatellite stable tumors, but they may be less responsive to chemo. So these are some of the MMR genes. So MLH1 and MSH2. These ones are abnormal in about 90% of hereditary non-polyposis colorectal cancer or Lynch syndrome. MSH6 is much less commonly seen in that, 5 to 10%. And MYH is a DNA repair gene. It's found in some cases of APC mutation that are negative for FAP, and they also increase the risk of colorectal cancer. So let's talk about epigenetics too. So we kind of just went over a lot of the different, proto-oncogenes, tumor suppressors that are often implicated in colorectal cancer. But epigenetics is a large area of research, and our understanding of how this affects our cancer risks is growing as well. So CPG islands are commonly found in gene promoters when hypermethylated downstream genes can be silenced. And so this can lead to, even if you have the normal genes and you don't have a germline mutation, it can still silence these genes so that you're effectively deficient in them. So this is a mechanism of action for many sporadic MSI high tumors. So typically MLH1 is silenced versus germline mutation as in Lynch syndrome. So the CPG island methylator phenotype, or CIMP, is found in many sessile serrated adenomas as well. And these will typically harbor BRAF mutations as well. So right versus left colorectal cancer, there's typically distinct embryologic origins and microbiomes. Right-sided is more likely to be hypermutated and in elderly patients. Left-sided is more common in young patients. The new ASCRS textbook, the 4th edition, just came out. Some of this is from there. There's a lot of increased understanding from recent research and literature in these fields. So I think that, again, our understanding of how all this interplays to affect not only just colorectal cancer in general, but specifically rectal cancers, right-sided versus left side, is growing. And we'll find that this potentially will influence our treatment patterns in the future as we understand more and more about what specific processes lead to cancers depending on where they are. So let's talk a little bit about young onset colorectal cancer. So it's increasing incidence of colorectal cancer in patients under 50 years old. It's predominantly left-sided, particularly rectal. This is due in part to this. The ACS and multiple other societies have decreased screening age to 45 years old. The NCCN has not yet changed their recommendations, but in general, this is what I'm using now in my recommendations to patients. And of course, this is for asymptomatic patients, so patients who are there for purely screening purposes. And I've not really found many issues in insurance companies denying colonoscopies in these patient population. Assessment and treatment is similar, but they're more likely to require urgent intervention. Genetic testing is currently recommended, as about 16 to 20% will have some heritable form of colorectal cancer. And 75% of these patients will not have a first-degree relative with colorectal cancer. So you shouldn't allow that to kind of drop, the suspicion of a possible hereditary colorectal cancer from your radar. And this is just showing some of those trends of colorectal cancer incidence. You can see age 0 to 49, male and female, has been going up since the mid-90s. And this is counter to what we're seeing in patients over 65 and 50 to 64. So it's really the opposite trend that we're seeing in younger. Is this because we're screening more? I don't know. It's still kind of to be determined exactly what the cause of this is. So let's talk a little bit about some of the different hereditary syndromes. So there's multiple hereditary syndromes. They kind of split it off into generally polyposis versus non-polyposis syndromes. So the polyposis